We will get into this in depth in the other classes, you guys. So the nutrition class, supplementation class, peptide therapy class, and the epigenome and genome class will go over this stuff more in depth. But this is just an intro into more or less what we're going to talk about in the other courses and kind of the basic things that people need to know about nutrition and supplementation. So this is something that very, very few regular doctors understand. The DNA, so DNA is a double-stranded helix in the nucleus of our cell. The DNA and RNA don't really do much on their own that we know of right now in the human body. They need to get translated into proteins, and the proteins are the stuff that actually do stuff. Like proteins are enzymes and cytokines and all those things. Those are proteins. So a double-stranded helix of DNA gets translated into a single strand of RNA, and then that single strand of RNA gets translated into proteins and peptides. So it's like translating English to Spanish to Chinese. If any malfunction happens in any of those translation processes, the end product is going to be faulty and it needs to be recycled. It'll be gobbled up and the whole process will go over again. The problem with it, with screwing around with RNA and DNA technology in humans are many. It's absolutely insane to be messing around with this type of RNA and DNA technology right now. We're hundreds of years away from understanding this stuff properly, if we ever understand it properly to start screwing around with it. So depending on the research that you read, 90 to 95% of our DNA, we have absolutely zero idea what it does, meaning that 90 to 95% of our RNA, we have zero idea what it is. If you guys have heard of the junk DNA that we all learned about in school, what we didn't learn about in school was that's 95% of our DNA. Only about 5 to 10% of our DNA encodes for protein. So that goes from DNA to RNA to protein. 90 to 95% of our DNA does not code for proteins. So let me just repeat that so people can understand this. We do not understand what 95% of our DNA and RNA do in our body. We know that if you remove it, you die, so it's obviously important, but we have no idea what it does. So to be messing around with these processes in the body is absolutely insane. On top of that, the epigenome, which we'll get into in 1 second, and it's much more discussed in the epigenome class, the epigenome can be inherited. So these are changes that you can make, markers, tags that you can add to your DNA that can actually be passed on to one generation from the other. So if you have your RNA and DNA screwed with as an adult, that is likely to be somehow passed on to your offspring. Very, very dangerous stuff. Not good. Here's, again, just another relatively easy way to look at how DNA gets read into proteins. So we have our chromosomes in the nucleus of our cell. We have mitochondrial DNA from the mom. The double-stranded helix gets translated into a single strand. So double-stranded DNA gets translated into a single strand of RNA. That RNA gets translated into a protein. The proteins are actually things that do stuff. So they're made up of different codes. You get different letters for the DNA and the RNA. Again, all of this stuff will be gone over in depth in the epigenome class. But this is why, you guys, this is why eating well and supplementing well works, and it's also why eating poorly and not supplementing is bad for your health. The reason that eating good food does good things for us is because it changes the epigenome, which is a level above our DNA, our genome. So the epigenome controls how our genes are read and expressed. So if you're eating food that your body doesn't like, it's going to alter the epigenome, and it's going to change the way that our genes are read and expressed, or if they're expressed at all. So there are certain gene anomalies that many people have. They will get expressed if you eat certain processed foods, and they won't get expressed if you don't. So that's the difference between health and sickness. Here is a good example of protein folding. So I think a lot of people have heard about protein folding. So the proteins in DNA need to get folded inside the cells so they fit everywhere. And then they get unraveled to be read and to go out and do stuff. So the left picture is just kind of the primary, secondary, tertiary, and quaternary structures of protein. The picture here, you guys, this is the process of what happens when our DNA to RNA to protein transcription and translation goes awry. So what protein a certain gene is going to produce totally depends on the environment, the cellular environment that the cell is in. Each gene can produce lots and lots of different proteins. It only produces the proper protein if the cells are in the proper homeostatic environment. If our cells are too acidic or too basic, or we have chemical concentration abnormalities, the proteins that are produced by the DNA to RNA to protein transcription and translation process are going to be faulty, misfolded proteins, like the alpha-synuclein protein that we see in the brain with neurodegenerative disorders, or the beta amyloid plaques. Those are misfolded proteins that our body has not been able to gobble up. So if you have enough ATPs and your mitochondrial health is sufficient enough, when our body makes misfolded proteins, it recognizes it, it gobbles up the proteins, it throws them out, and it starts the process over again. The problem that people have with things like Parkinson's and Alzheimer's and COVID and all this other stuff is that if our body is in a state of energy deficiency, which again, 80+% of the country are in a state of chronic energy deficiency, our body only has a finite amount of energy to use. If we run out of ATPs, then our body and our brain has to start making hard subconscious decisions of where to send that energy. But the bottom line is there's not enough energy to properly recycle or clean up the garbage, more or less, in the cells. So as we become energy deficient, our cells are in a worse environment. They start to misfold proteins more, so the translation or the translation from DNA to RNA to protein malfunctions more. In a healthy state, it would malfunction less, and we would have the energy to gobble up and throw out the misfolded proteins. As we become energy deficient, we have more misreadings, we have more misfolded proteins, and we run out of energy to recycle those misfolded proteins, so they start conglomerating in nerves and in nervous tissue, and that's what leads to things like the beta amyloid and alpha-synuclein plaques that we can see in Parkinson's and Alzheimer's disease. One really, really cool study, and this is just a good example of how little we actually know about the human body. About 5 years ago, a group in France found the alpha-synuclein protein that's related to neurodegeneration that we typically see in the brain. They found that protein already misfolded in the intestines. This has never been seen before. So what these researchers did is they radioactive biomarked the misfolded proteins. Those misfolded proteins traveled up the vagus nerve and ended up in the brain. So now we know that there's definitely numerous types of Alzheimer's and Parkinson's. There's some that start in the gut, there's some that start in the brain, and I'm sure there's other pathways that those misfolded proteins end up in the brain, but they don't all start in the brain. So this is just another good example of how complicated our body is, and to be messing around with our nuclear material like DNA and RNA, it is impossible to say that that is safe for anybody, because again, we don't know. The government says this on their website. We have no idea what 90 to 95% of the DNA is responsible for. Therefore, we have no idea what 90 to 95% of the RNA is responsible for. We have no idea what we're doing with it.